Hello, everybody, and it's a pleasure to see you around. I'll try to cut my talk a little shorter, so Ruth will have the time she planned to talk. What I'll be trying to do now is kind of walk you through the next stages. So we've heard about genome assemblies, we've heard ideas about pan-genomes, but really what can you do with the pan-genome are done? What's the next step after that? So I'll try to walk you through what NRGene's vision is around that, and what are the practical steps or the things that are already happening, and even some of you might think of joining them. So it's really one big system from NRGene's point of view, and I'll walk you through the different parts of it. But it kind of separates into building genomes, which we've seen examples of, building pan-genomes, which NRGene has a very unique approach to, and then using the pan-genome to build haplotype databases that are then used to capture diversity and really design, I would say, the most optimal genotyping for different uses. The main use will be, of course, breeding. So that's kind of a high overview of the product line, as we call it. So it's mainly 3 different parts. The first one you just heard about is the assembly. Then there's a part that takes several fully assembled genomes and compares them together on a sequence level, so chromosome to chromosome level, and then compares the gene space. And this is actually tunneled or used as an infrastructure for next phase, who will actually, I would say, analyze a larger diversity, much larger diversity. And this is where we're coming into really covering a diversity big enough for breeding. And the idea is to cover that in a way that's, one, cost-effective, and the other, it's actually translatable to a breeding settings with very efficient and high accuracy genotyping. So we had a talk today with some of our collaborators, and I mentioned that 3 years ago I was putting this vision that genomes should be basically like a reagent. Right? You need the genome, you should get it. It shouldn't be a big deal. And I think we're slowly getting into this point where researchers don't feel anymore that they need to really make a business, like a big project out of assembling a genome, but rather, if they need genomes, they can get them and move on to the biological question behind, or that is answered by those genomes. So this is where we're going, and now how to actually get there. Okay, so this is the de novo assembly. I'll kind of jump over it because you heard so much about it. I want to kind of address some of the points that were unique to the last year. So in the last year, on top of what Niels has shown, we've done additional assembly projects, and many of them were heterozygote, even complex polyploid heterozygote genomes. And again, this is, I think, the first time that those communities of the strawberry, of the sweet potato, and the others can actually have a reliable genomic assembly to actually use. And now they can think about the next steps. Okay, so what is the next step? What we, as in NRGene, define as a pan-genome, and that's kind of, I think, some of the questions here were pointing to that, is that you really have to have fully assembled genome chromosome-wise, and then you're actually mapping those chromosomes to one another from a whole set. So it's an all-to-all comparison of a set of genomes, okay? And then you take a set of transcripts. Now, the set of transcript, as many people mentioned here, is not trivial. So NRGene does not do the annotation part, but once the annotation is ready, NRGene will take the annotation and compare the whole transcript sets between the different genomes. And what do you end up with? You end up with 2 main things. Both of them are very useful. One is the mapping, and this is related to all those structural variations that you see between the genome. So mapping coordinate-wise between the different genomes, all to all. And the next step is the understanding of which of the transcripts are actually matching, copied, deleted, or translocated between the different genomes. Okay? So this is what NRGene is defined as a pan-genome. This collection of data, the mapping and the gene space mapping, is what NRGene is defined as a pan-genome. What can we actually do with it? So before I'm going into the actual usage of it, let's look at how many of them are going to be around. So we have started several, or it's not exactly us, we are supplying the technology for groups of combined academics and commercial partners that each one is concentrated on a specific crop. And here is a call to the community to join in such efforts, because I think they're really changing the way that the genomic diversity will be analyzed in the future. Ongoing projects that have actually met here in this PAG and are looking for additional partners are the potato pan-genome, the barley pan-genome, as Niels mentioned. The wheat pan-genome is at the end of basically the work. There's a cotton pan-genome being arranged. There's a canola pan-genome being arranged. And hopefully the others will be announcing that they are in progress soon. If any of you feel that he would like to join such a consortium, and such a consortium will end with a publication of the pan-genome. Okay, so this is not an industry effort. It's an academic effort at which industry contributes to, and the end will be a publication of a pan-genome. You're very welcome to approach us, and we'll connect you to the key opinion leaders that are actually leading those pan-genome efforts. Okay. What's the next step after pan-genome is there? This is a very quick glimpse at the pan-genome visualization. Okay, so for example, if you use one genome as a reference, one genome out of the pan-genome as a reference, you can actually map the other ones. Okay, so if this is the genome, all those tracks underneath him are actually mapping the other genomes in the pan-genome to this genome. And what you actually can see, each rectangle here is representing a gene. You can color code the existing or the non-existing genes on the other genome. So the fact that this rectangular is green is indicating that in this genome, that gene appearing on the reference genome here is not existing, and the reference genome is a random pick here, right? So each one of them can be the reference genome on the kind of reflecting picture, mirror picture of what I'm showing now. Okay, so it's the transcript themselves and the differences between the content of the genomes. So I'll quickly kind of scan to the following slides, because I've shown them many times. What I really want to focus on is why is the pan-genome so important. Okay, so instead of aligning short reads to a reference genome, which is what most methods do, we align short reads to themselves first. So we assemble short reads together from each genotype, and then we align the contigs to the pan-genome. Okay, so that really gives you a much better way to capture diversity. We're dealing with long, phased, and clean contigs to align to a pan-genome, which capture a lot more of the diversity. And then, when you're actually looking for those subsequences that are unique, let's imagine those contigs are aligning in the same place in the pan-genome, you can really compare them and find all the differences. So what we do is we kind of run all this to a big data machine, and we extract the subsequences of all the contigs that are unique, and we hold them in a database, creating this kind of graphical picture with maize, for example. So this is the 10 chromosome and similarities and differences color-coded between the different genomes. But what we can really do is actually genotype by them. Okay? So we build this haplotype database. We keep all those millions of markers within the database, and then we can scan a much larger diversity just by random sequencing. And we believe this is the future of sequencing or genotyping by sequencing. So it's as cheap as possible, random sequencing that you can use to reconstruct the haplotypes, not the specific SNPs. each combination of reads will be different every time you run it. But there'll be enough reads to actually construct back the haplotype and read back this block from the original database. So this is representing sequence block, and now you're reading back those sequence block based on that sequencing data, okay? And then you can actually do that again and again, completing your haplotype representation. So I'll just finish by, this is the heterozygote case, which I won't get into it, but it exists. And what I really want to announce here is that NRGene is going to offer an additional optional service, okay? Starting with DNA, and instead of doing what we call assembly or a diversity analysis, we'll basically impute back, okay, from your current genotyping mechanism. We'll compare it back to the haplotype database and end up with a haplotype level information genotyping. So for wheat, for barley, and all the other potato, it's very soon to come that you will be able to take either your current genotyping or your current sequencing-based genotyping data and basically move it or read it through the database to get a much higher resolution and a lot better genotyping data for really a very cost-effective price. So this is what NRGene is working on right now. This is going to be rolled up crop by crop in the coming years. Ruth, I hope it didn't take too much of your time, and I'll take questions.